Okay, ladies and gentlemen, I apologize for not being able to record this on Zoom. For some reason, Zoom wasn't working when we were trying to get this review session started. So I'll be recording this via the camera on my computer. So at least you can have the voice aspect of the review and then sync it up with the slides as you go through them. If there are any questions, please feel free to email me during finals week. However, just know that it might be at max 24 hours before I can get back to you. Um, we're all fairly busy, and I have a couple exams myself during finals week. So please let me know if you have any concerns or questions, and we'll get started with this review here. Okay. So, final exam here. We have, let's see, I do this. Oh, yeah. Okay. So this is the format. It's 100 questions. There's no matching. It's only multiple choice. I just finished making them all multiple choice. It is open note, not open AI, not open Google search, just the slides and your notes from lecture. That is it. Notes from lecture and slides. And the answers will not be shown until after the semester is over. So there's no hope in trying to get your partner to cheat for you. Let's see. Basically, the best thing that I can do for you guys to review is go back and look at my other review slides for exam 1 and exam 2. These are some of the highlights from the exam on something that would be worth studying. However, some of these things are much more in depth than just this one line on the screen. Synthesis of ATP through pathways of the cell. I mean, we talked about the Krebs cycle, fermentation, glycolysis, and all that stuff. So there's a lot on that. But I would find it worthy of review to go and find these bits of information. Any questions about that? No questions.
Well, is there going to be a lot on metabolism? Like that first test where it's just like a bunch of how many ATPs produced and stuff like that?
I would not say that there is that style of question on the exam. I would still find it very useful to study metabolism, knowing the processes,
Okay.
especially with our most recent applied microbiology, where we were talking about like cheese and alcohol and things. Those are all fermentation processes. So knowing like what those do, that'd be good. Anything else? Okay. All right. So chapter 21. I also in later slides I included which slide deck they're from, so it's a little bit easier for you guys to find. Um, the learning objectives for this one and for all of the bacteria are really just knowing the disease, the transmission, the treatment, and the prevention. If you got those four things down, then you're good to go. Disease, transmission, um, like how it's transmitted, treatment, and prevention.
Testing out, is everything like generally in order that you learned it, or is it all jumbled?
It is all jumbled up. So the test is it goes from one topic to the next, and you might have an exam 1 topic in the middle of an exam 3 topic. Yeah, it's not exactly straightforward, which kind of makes the whole 2-hour time limit even harder. Yeah. But anything with a chart, as you can see on this slide, if it's green, it's good to know. Green is good. So knowing that our rickettsia is going to be with wood ticks and hard ticks, and that's where they're going to stay, and they're going to give you Rocky Mountain spotted fever, or SFR is the short form. And knowing those kinds of charts would be helpful. Okay. Also knowing how rickets disease is treated is a good idea to know. Okay, chlamydia. Lots of information on chlamydia. good old clap on. Yeah. Again, disease, treatment, prevention, and transmission. If you know those, that helps a lot. A couple other things that I made sure to highlight in here was that it's classified as an STI, and that the bacteria that causes this disease um, has two membranes but no peptidoglycan. So there's no PG layer in them, but um, they're still a pretty hard thing to kill. So, any questions about chlamydia? Good. Hopefully, you never need to know about it. Okay, life cycle of a tick. That kind of goes along with the rickettsia and the rickets disease. Um, Treponema would be good to know the different forms of it. Treponema pallidum. Talking about where it's coming from as well as what it causes. Pinta, yaws, Begel, syphilis. Knowing where those things are, and then also knowing the course of untreated syphilis would be helpful. Uh, Again, any kind of chart that he throws in would be good to know. Pictures of diseases, not so much. You'll not really have to identify diseases that way unless you're a doctor. So that was kind of the last of the little. I think got one more. Nope. Okay. So that was the last of the bacteria. Chapter 21, that kind of rounds it out from what we didn't cover on exam 2. And now we're going to be talking about viruses. So on this next slide, on chapter 13, we're going to be talking about the general overview of viruses. And then that's slide deck 33. And then we'll be getting into different kinds of viruses, RNA viruses, DNA viruses. Any questions about bacteria before we hop into viruses? Okay. Any questions about viruses? All right, cool. Sorry, it's not exactly like colorful or nice to look at. It was kind of made in a hurry last night. Uh, Viruses, knowing everything on this slide would be good. Yep. Can anybody tell me the difference between double-stranded DNA and double-stranded RNA?
Like chemically, or like what happens like to the virus?
Any differences?
It's U instead of T.
Right.
No.
So there's uracil instead of thiamine. I can speak. Yeah, that's the biggest difference. The way I always think about RNA viruses is it's faster to upload into a cell's genome or ribosomes, but it's unstable because it's got to fight through the cell to get there. Whereas DNA viruses are a little bit more stable because of the genetic structure of it, the chemical structure of it. But they're harder to translate and transmit. Single-stranded DNA is going to be easier than double-stranded DNA. And just know that there's only one double-stranded DNA that we care about in this class. And I believe there's a chart that outlines that. Any questions about cancer or prions or viroids or viruses and cancers? What's an example of a virus-caused cancer?
Isn't papillomavirus?
Yep, yep. HPV counts. Yep. So warts and things. That's why you should wear shower shoes and stuff if you're showering with different people.
The human herpes A we were talking about?
Yep, yep, yep. So uh, HHV 8. That one's the one. Good. Cool. Okay. Chapter 24, slide deck 34. This is going to be talking about DNA viruses, DNA virus families. Right. So we have our double stranded DNA, the single stranded DNA, and there is one which is a mixed. It could be DNA that is double stranded or single stranded, and that's going to be the hepatodaviridae. Say that 10 times fast. Um, really, the most common distinction, I know we kind of talked about herpes viruses today a little bit, um, but the major distinction is going to be where it exists. If it's genital herpes, it's going to be in that sacral region, and if it's going to be oral herpes, it's going to be um, near the mouth or really anywhere else. There's a couple genetic differences, but we don't really worry about those. Where does herpes hide out? And
The capsid.
nervous tissue. Uh-huh. Yep, nervous tissue and B cells. Yep. So your own white blood cells carried around. Let's see. What kind of that's a hard it's an opportunistic virus. Just to be clear, it's when the host is weaker in any shape or form, whether that be physically, mentally, emotionally, or whatever. Any kind of stress, um, the herpes virus can rear its ugly head. Gone. Chickenpox. You got chickenpox and shingles. Not fun. We have had we've had a varicella zoster virus vaccine for a long time. Have you guys received that? Actually, I can't ask that. Never mind. Legally, I cannot. But I have. And it basically just preps you for it, lowers your chance of developing symptoms. And then pox viridae, which is different than chicken pox. It's smallpox. And the MMR vaccine, that's going to be pox viridae. Any questions about this slide before we move on? Okay. All right. Epstein-Barr virus. Yep, knowing the diagnosis, treatment, transmission, and prevention would be helpful for that. They kind of work in the same way where herpes extends, uh, where they hang out in B cells and can just kind of wear them as a coat to get around the body. It's like a little invisibility cloak. And then cytomegalovirus. Uh, yeah, any questions about these viruses on there? Okay. Uh, One thing that is not on here is how these guys are transmitted. How do they get from person to person typically? You guys know? Okay, that'd be something good to look up. Let's see. Yep, all of these are great charts to kind of help you guys figure out um, which causes what. Especially this one here. All of these guys. These are all positive ssRNAs. Can anybody tell me the difference between a positive and a negative single-stranded RNA? Mm-hmm. Right.
It has to be translated to positive.
Right.
Make it replicate.
Right. And what does that exactly do in a cell? Like, does that make it better or harder to fight or?
Negative sense it has to be like flipped around because it is the starter that goes from the 3' to 5'. It's supposed to be 5' to 3'.
Uh-huh. Yep. So the starter is on the wrong end. So it's got to be flipped over before it can start replicating. And that's a problem for the virus, but it's better for us. This is because we can actually, we're okay. We're better at finding those ones because it takes them a little while. If you think about it, these ones up here, these encephalitis, the Japanese West Nile, St. Louis, those yellow fever, dengue, pretty common. We've probably heard of them before. Whereas these like Rift Valley fever, sandfly fever, or Congo hemorrhagic fever is not as common. And we kind of don't really hear about those. Okay. Chapter 25, slide deck 37. This is all about HIV. And then here's your little diagram down in the bottom left corner, where's the replication of retroviruses between single strand. Excuse me. Single-stranded RNA and negative sense single-stranded DNA and double-stranded DNA. And what we're looking for is how they interact with tRNA and how they kind of look into in the ribosome for being able to transmit themselves. DNA is not stable on its own, so when you have single-stranded DNA, you kind of got to pair it up with something else. So you kind of have to have another piece of RNA just to stabilize it a little bit, but it still works in the same capacity. Knowing these diseases, like bacterial infections, would be tuberculosis or rectal gonorrhea or like salmonella or streptococcus. Fungal infections is going to be pneumocystis, pneumonia. I wouldn't really focus on fungal protozoal or the others section, but I would absolutely look at the bacterial infections, the viral induced tumors, and viral infections. It's kind of the majority of what we covered in class. And then up here in the top left, we have the progression of HIV. So clinically, this is what the time frame of an HIV infection looks like. And usually, uh, the person who has HIV does not pass away due to HIV itself, but through other diseases. Like the common cold can be a serious problem if you have HIV, where it wouldn't really be a problem to a healthy individual. It would be a pretty darn problem, pretty big problem for you. Question about this? Okay. Here's your list of single-stranded positives and single-stranded negatives for um, DNA viruses. This is slide deck 38, chapter 25. And then their appropriate viruses over here from the right. Coronavirus was a single-stranded positive RNA. It's helical. Knowing what helical and like icosahedral is, what that looks like. That's just a helpful visualization. And then down at the bottom here, we have the rotavirus and the Colorado tick fever for the Reoviridae. That is your double-stranded DNA. It's your double-stranded DNA virus. So, good to know for the double-stranded DNA viruses. If I can make that any more clear. Colorado tick fever as well as rotavirus. And then also knowing if these guys, what is the difference between a virus being naked or enveloped or enveloped?
Doesn't it have like a shell on top of it?
Right, and what's that shell made out of? Uh huh.
It's like a
It some of them have that, not all of them, but the shell that you're thinking about is going to be the glycoproteins, right? So that sugar protein capsule. And then a naked virus is not going to have that, but still is going to have a capsule around it. So the envelope viruses are a little bit harder to uh, kill, but are easier to track uh, because they have an extra layer on the outside to protect them. Cool. Don't worry about the size. Uh, just know that they're really freaking small. Okay. Okie dokie. Oh, we're close to the end. So slide deck 39, chapter 26 is going to be talking about the applied microbiology, as well as slide deck 40. The applied microbiology is all about that cheese and that alcohol and the wine and the beer that we talked about, as well as the antimicrobials and antibiotics kind of vibe. So like D values kind of come into play here. Wink, nudge. Um, any questions about applied microbiology? It's not a very large section. Okay. Let's see. Okay, drinking water and how that is filtered. So we have treatment plants that we can find. We also have wetland filtration. We also have just like there's a whole bunch of different methods, but the traditional way of water treatment would be following those four steps there. Sedimentation, flocculation, filtration, and disinfection. Um, we do a variety of methods here in Utah for disinfection of the water because we get all of it from our reservoirs. Specifically in Provo, we get most of our water from the Deer Creek Reservoir, and it's open to everybody. People swim in it, boat in it. I don't want to drink motor oil or somebody's pee, so we disinfect it quite a lot. um, let's see. Everybody knows about anthrax, right? Uh, Ebola. Let's see. Any questions about possibly calculating BOD or knowing what that means? I think we had a couple slides on it. Yeah. It's pretty simple. If you have a lower BOD, the biochemical oxygen demand, we can use that in two ways. One, to calculate how oxygenated the water is, and two, to calculate how many bacteria, aerobic bacteria, are in the water um, or in the waste, right? So if it has a lower BOD, that means there are less aerobic bacteria in the water, which means it's usually better for you. Less bacteria is usually better. Okay. I think the next slide is the last slide. So any questions here? Alrighty, righty. This whole slide is green. And like I said, very important to know the disease, how that disease is transmitted, how that disease is treated, and how that disease is prevented. Cool. Questions, comments, concerns? I think that's it. Yep, that's it. Okay. It's fairly short, so hopefully it's not too bad. If there are no questions about this, are there any questions from class today that we want to talk about? Anything that we want to review for the exam? Anything like that?
So basically, like the exam 1 and exam 2 is pretty good to study.
Yeah, yeah. If it appeared on exam 1 and exam 2, I would highly encourage you to study those kinds of topics.
Like, study the topics.
And the quizzes too, or just the test?
Prep quizzes are pretty darn good. If there's a thought question in the slides, that'd be good to know too. Knowing the lecture quizzes, I know towards the end we kind of got a little lax with the questions, but going through those lecture quizzes helps. I think you're able to see those. I know for sure you're able to see exam 1 and 2. So you're able to go back through those and look at your right answers and your wrong answers. Yep. Yeah, like he said, the majority of the test is on past information. I think it's about 35 or 36 questions on the most recent stuff, and the rest of it is all on the old stuff. So in total, 100 questions. You have 120 minutes to take it. I would not find it very useful to go hunt for every answer. So make sure you know your stuff before you take it. Yeah. Yep. Good luck, guys. You're gonna do great.
Thank you.